Hello guys. Um, Today we are going to discuss the free living amoebas. Uh, we have discussed some of the amoebas before, including these intestinal amoebas like uh, Entamoeba, histolytica, and others. Today we are going to start with the free living amoebas. Uh, their importance, and the different conditions that these amoeba are causing in human beings. So first, we would discuss Naegleria fowleri, and after that, we would discuss Acanthamoeba and other species as well. Uh, so, uh, Pathogenic free-living amoebas that we are discussing right now include different species from different genera, including genus Naegleria, the species Naegleria fowleri that we are going to discuss today, genus Acanthamoeba. There are different species in genus Acanthamoeba, as you can see here. Genus Balamutia, and the species that are causing problems that are pathogenic, and Sapenia diploidea. So the pathogenic free-living amoebas means amoebas that are living uh, free, and they are causing different types of diseases. What does free means? The free means that they do not require any host. Uh, for Usually you would have seen that parasites require a host for living, right? Because they are living at the expense of their host. They are getting nutrients from their host, and the host sometimes would get some benefit out of this relationship, other while sometimes it may not get any benefit out of it. So these are different type of host parasites relationships that you would know about. But pathogenic uh, parasites always cause diseases. It means that they are harmful to their hosts. These are parasites, these free-living amoebas, which do not require any host. So you should keep that in mind. And the second thing that you should remember is that they do not require any vector for transmission into human beings or animal species. Naegleria that we would discuss in this lecture also causes problems in animals, not only in human beings. Okay. There are different experimental models which have been successfully infected with Naegleria, indicating that it can be equally harmful to these animal species as well. So no vector is required for transmission, as you would have seen some parasites which are using very difficult strategies to survive. For instance, they would need two different hosts to multiply in. So it is sometimes very clever of them, or it is sometimes very difficult for the parasite to manage that, to bring two closer hosts together in order to complete its life cycle. And you have many examples of that. For instance, if you take the example of Plasmodium. So Plasmodium, on one hand, is replicating in a mosquito, and on the other hand, it is replicating inside the human being, which are two very different creatures. So it means that a mosquito with a very simple makeup or simpler genome as compared to the human, and with a very different physiology and anatomy and everything. You know, it the parasite is completing some sexual stages of its life cycle inside a mosquito, and then it jumps to the human being. It needs a human being. So for this kind of relationship uh, between the host and the parasite has to do a lot in order to survive by uh, getting two hosts together in some relationship in order to keep this its own existence going. So in this case, in the case of pathogenic free-living amoebas, they do not require any host. They do not require any vector for transmission. Uh, uh, they uh, are not well adapted to parasitism. Other parasites, they are well adapted to parasitism. They parasitize on other organisms or hosts. But in case free living amoebas, they are not very good parasites or other. They can live freely in water bodies, and uh, they eat on a different kind of bacteria inside these water bodies, so they can survive freely. But they are different from bacteria and other organisms. They are protozoans, they are unicellular, these cells are eukaryotic in nature, they behave uh, like full-fledged organisms, and they can live independently inside these waters. So these are parasites which can live freely, but which can also infect human being and animal species, uh, which they do occasionally. Uh, the Naegleria fowleri, it's also called the brain eater. The brain eater parasite. Um or a brain-eating amoeba. It causes an acute and fulminating primary amoebic meningoencephalitis, or inflammation of the brain tissue, or the covering of the brain tissue. Here you can see this is brain. You can say at the right bottom corner. You can see this is the image of the brain after uh, Mm, it was infected with Naegleria fowleri. So after the infection, you can see these patches, red patches, and necrotized brain tissue that has been caused by uh, this particular parasite. You know, so it's a brain-eating uh, parasite. It was discovered in the 1960s in Australia by a doctor whose name was Dr. Malcolm Fowler. This Fowleri also is after his name, the discoverer of this particular parasite. So he discovered it in Adelaide Children's Hospital. Um, and he described several cases of this particular parasites, which was prevalent in that setting and causing problems in children. This is the image of Naegleria fowleri, and it is found in warm fresh water in lakes and rivers, inside hot springs. Also, it is found in pools which are not chlorinated. So you will not find them in chlorinated pools, but inside unchlorinated pools, you can find lots of these Naegleria fowleri. So it's amphizoic, which means that it can survive in a free living state in water, soil, or in the host. which can be in the human central nervous system. Uh, if you look at the morphology, Naegleria fowleri, you can find Naegleria fowleri in two stages. Stage 1 is the trophozoite stage, and stage 2 is the cyst stage. The trophozoite stage again is divided into two stages. You can either find the trophozoites in amoeboid forms or in the flagellate form. So what are these two different forms? You can just look at this form. It's a trophozoite. Both of them are trophozoites. What is a trophozoite? It's a vegetative or feeding stage of the amoeba. Uh, Found in CSF or in the tissues. When you do some diagnostic procedures to find out, uh, you know, uh, the causative agent for meningoencephalitis, then you take out sample of the cerebrospinal fluid from the back of an individual. You just puncture it with a needle and take out the CSF. And then you test it for this particular pathogen. And it measures about 10 to 20 micrometer in diameter and has granular cytoplasm and a distinct ectoplasm. As you can see here on the right side, this is amoeboid form of this particular parasite, the trophozoite form of the parasite. So you can see the nucleus. Inside the nucleus, there is a nucleolus. There are very distinct mitochondria for energy production, you can say. There are contractile vacuoles. There are pseudopodia here on the back. There are fodovacuoles, and there are also lobopodia. These are lobopodia. These projections, these are lobopodia, which are used for attachment or ingestion of bacteria. When it's going to attach with something, or when this amoeboid trophozoite is going to trap a bacteria and ingest it, then it uses the lobopodia. So this trophozoite, it's a vegetative and feeding stage of the amoeba, and it has well characterized structures. If you look at the structures found in this cell, you would find out that it has all the specialized structures to carry out different jobs. It's more like an animal than like an individual or single cell. The second form of the trophozoite is the flagellate form. You can see these well-developed flagella, two flagellas that you see that are attached to the basal body, and there is an apical nucleus, at the apex or at the interior position. You can see the nucleus here and other structures which are present there. The flagellate form is elongated and pear-shaped, as you can see. There is anterior nucleus. Anterior means at the cranial side or towards the head of or the front of this particular pathogen. You can see the nucleus, which is here at the apex. And there are two flagella. This is the trophozoite form, flagellated form of the trophozoite. It's often encountered in water bodies with 27 to 37 degrees Celsius temperature. It's a non-feeding and non-dividing stage, the flagellate stage. Okay, the amoeboid stage that you saw here, it's a vegetative and feeding stage, and it's dividing state, whereas this one is non-dividing. And this is the flagellate form is never found in tissues. In the brain tissue, you cannot find the flagellate form. Can be transformed to amoeboid form? Yes. These two forms are interchangeable. So this kind of interchanging, between these two forms, or the cyst form, which is the third form, usually takes place in the case of this amoeba. Amoeboid form, this is the infective stage, okay, and amoeboid form transforms into the flagellated form. Flagellated form after some time can transform back into the amoeboid form. The amoeboid form similarly can change into a cyst form when the conditions are very challenging for the survival of the parasite. And when the conditions become relatively milder, then the cyst would uh, um, would become amoeboid trophozoite again. You know, so this thing just goes on and it depends upon uh, the conditions that are prevalent in that particular setting where the parasite is living. Now, the cyst is a resistant form of the parasite which offers protection from desiccation and shortage of food. And the function of the cyst here is similar to other cysts that we have discussed in case of other organisms. It's always the cyst formation, you know, takes place when the conditions are challenging for particular microorganisms. Then it's a survival strategy of the microorganism that is in place when needed. It measures about 7 to 10 micrometer in diameter and is surrounded by a smooth single-layer cyst wall. It has got a single nucleus, as you can see here, and a contractile vacuole and food vacuoles. All these things are there inside this particular cyst. And the cyst form also usually is not seen in clinical specimen. It means that if you get a clinical specimen for examination, like the cerebrospinal fluid or the brain tissue, or and you want to examine it for the pathogen, whether the pathogen is there or not. So you can find the trophozoite amoebite form of the pathogen or a Naegleria there, but not the cyst form or the flagellate form. They are not present in this. So if you look at the life cycle of this parasite is quite simple. Amoebite form, and it can just uh, divide and make other amoebite forms, or it can uh, go and transform into a cyst form when needed. Okay, and the cyst form again can be changed into the amoebite form. Similar is the condition with the flagellate form, as we discussed that these are interchangeable. One form or one morphological form is changing into another morphological form, and the other morphological form is changing back into the same morphological form. Uh, um, and this thing just goes on and on. And during this process which is going on, sometimes the human beings, they would be accidentally infected with the amoebite form. The amoebite form can cause infection in human beings, amoebite trophozoites that we are talking about. Sometimes human beings who are, taking a bath in some ponds, freshwater bodies are warm. In warm season, in warm season particularly, this thing has been encountered in different countries. So while taking bath in these water bodies, human beings can come in contact with this amoebite form of the trophozoite, and the trophozoite would then go and start eating on the brain of particular individuals. Uh, and they cause this condition which is known as PAM, primary amoebic meningoencephalitis. which is also called Naegleriasis. This condition is a very fatal condition, and swimming or diving in warm water or aspiration of water contaminated with Naegleria fowleri, the trophozoite species, in summers can cause this particular problem. As in the very first slide, uh, we saw that lakes, ponds, rivers, and different water bodies can have this particular parasite, Naegleria fowleri, and while swimming in these water bodies, sometimes the water can enter into the nose, and through the nose it can just get access to your brain sometimes. So the parasite, if present in water, can get access to the brain and cause this particular problem. The trophozoite is neurotrophic. It means it loves the neurons or the neural tissue, uh, and olfactory mucosa. Olfactory uh, mm, mucosa is on the olfactory bulb is, you can see inside your nose, at the junction between the nose and uh, your cranium or the skull, where the brain tissue is starting. So you are able to smell different things because of the olfactory nerves that are involved in this particular process. Okay. Um, what are these cribriform plate and subarachnoid space? We would discuss these things in the next slide. This is a kind of plate which has got holes for nerves. The nerves pass through these holes, and the nerves are coming out of your central nervous system and passing through these, plates, the holes in these plates, which are known as foramina, and they are innervating different parts of your nose. Okay, and they are then attached with these receptors in your nose, and they are receiving signals. The receptors are receiving these signals, and the nerves are transporting it to the brain where these are being interpreted. Okay, so it's an anatomical structure, the cribriform plate in the subarachnoid space that I would show you. The primary amoebic meningoencephalitis, as I told you, it's a very fatal condition. And it's called fulminant. Why it is called fulminant? Because it is sudden and severe. Okay, its onset is sudden and severe. It can take some time, a couple of days sometimes, for the condition to develop. But people have noticed that even a little is a time span of 24 hours sufficient for the symptoms to appear in some patients. So it causes a hemorrhagic inflammation when the parasite gets into your brain, and it start eating on the brain, and in this way it causes necrosis of the brain. Okay, and so necrosis of the brain is the death of the brain tissue, or the death of the neurons, or the brain cells, due to eating by this particular parasite. Okay, so cell death is of different types, you know that. uh, it may be because of programmed cell death, or apoptosis, which is programmed there inside the cell. When the cell comes in contact with something like radiations, or it is injured to the extent that the cell feels that it would no longer survive. So in that case, the cell would initiate its program for its own death. So it's a kind of you can say suicide, uh, which is called apoptosis. But necrosis is a kind of killing someone or killing the cell. You know, no programmed for cell death is initiated here. So it's a different kind of death. The brain tissue is damaged and it's you know, necrotized, and while the brain tissue is being damaged, different parts of the brain, they are doing different functions. That's why the patients, they would you know, describe themselves as, you know, experiencing different kind of symptoms based on the location of the brain tissue which is being damaged. And amoeba is the only form detected in the brain tissue. Flagellates and cysts never found in the tissue or the cerebrospinal fluid, and that's already I have told you. So here we discuss the pathogenesis a little bit more. The water-related activities such as swimming, underwater diving, or other water sports can result in water entering into the nose. Okay, and amoeba penetrate the nasal mucosa. Once the water enter into the nose, it penetrates the nasal mucosa. It's traveled to the brain through the cribriform plate. The cribriform plate, you can say, is a plate or is an anatomical structure which is, you know, isolating the cranium where the brain is from the nasal cavity. So you can say that this structure is an anatomical barrier between the two. And amoeba migrate into the brain through the olfactory nerves, causing primary amoebic meningoencephalitis or PAM in healthy individuals. And when the amoeba reaches your brain, what does it do? It consumes astrocytes and neurons inside your brain. Okay. Normally, it does not consume astrocytes or neurons. It is not feeding on astrocytes and neurons outside the body, but is outside the body, it feeds on different kind of bacteria in the water bodies. But inside the body, once the mane is accidentally exposed to this particular parasite, it starts consuming these astrocytes and neurons. And the trophozoites in the in CSF and brain tissue, flagellated forms. Some people say that occasionally you can find in CSF, but many people, you know, say that only the amoeboid form is present there in the CSF or brain tissue. So, what is the cribriform plate? This is a cribriform plate. You can see here, you know, here and here. These two places, these are making the cribriform plate. And this is the olfactory bulb, the yellow one. This is the olfactory bulb. And this bulb contains different kind of mitral nerves, olfactory nerves, and these olfactory nerves are passing through these tiny foramina. If you see these tiny foramina are these tiny holes in the cribriform plate, you know. So the nerves, they are passing through this, you know, and this is, you know, uh, you can say the inner side of the skull that you are seeing here. This bone in the middle of the cribriform plate, which is dividing the plate into two, is the crista galli. And you can recognize other structures. These are sockets for eyes. This is socket for, you know, other structures. You can see these are different structures of the skull, actually. So you can say this is in, you know, the inner side of the skull. And on the other side, this structure is continuous with your nasal cavity or the nasal structure. Okay, so the nerves going through these structures would just enter into your nasal tissues, where they would innervate different parts of the nose, the upper, lower, and middle part. So during its pathogenesis, what does this parasite actually would do? Amoeba would produces cytophatic enzymes like hydrolases, cysteine proteases, phospholipases, and lysosomal enzymes. And it would carry out destruction of the neural tissue in the brain. Is the nerves are coming here, the olfactory nerves, they are coming, the olfactory nerves are going to the brain, taking the parasites along with them. So inside the brain tissue, these different kind of enzymes which are produced by the parasite would destroy the brain tissue. Okay, and in addition to these things, it's thermotolerance and the pore-forming proteins that it's making, and the complement resistance that further helps with the spread of this particular parasite in the brain tissue. And in this way, it damages the brain, necrotizes the brain, and is it is going to necrotize different parts of the brain, different symptoms of the condition, they would appear. And this is a brain tissue. You can see this brain tissue. And this is the necrotized tissue with a little bit deep color that you can see. uh, you know so this is from a patient with primary amoebic meningoencephalitis. Okay, so if a part of a brain which is controlling your locomotion is affected or being eaten by this particular parasite, then you would start incoordination, in your the moment, or other conditions, headache, stiff necks, and status changes and seizures and things like that, like hallucinations sometimes if some parts of your brain controlling your imaginations or these things is affected, then you would also experience such kind of symptoms during those days. And it happens in children and young adults who are fond of, taking a bath in these water bodies, and and people who have a history of bathing, swimming, diving, or playing in warm, stagnant fresh water. Uh, it takes about a week or two for the symptoms to develop. So that is the incubation period of this particular uh, parasites. The diagnosis: history of the swimming in pools or natural warm water. This is very important to ask about the history and the clinical picture which is there. The headache and these different neurological symptoms that appear over time. That is also important to know. Then you take the cerebrospinal fluid, you make a fresh film of it, and you look for the amoeboid parasites uh, in the cerebrospinal fluid. And there you would see increased pressure, polymorph nuclear cells, increase in protein, the presence of RBCs there in the cerebrospinal fluid. You can stain it. You can use JAMSA or right strain or trichome. You have different options for that. Or you can alternatively culture it on non-nutrient agar plate seeded with E. coli. And also leukocytosis in peripheral blood could be observed in patients who are infected. It's 25,000 or more leukocytes inside your blood. If you are infected um, with this particular cell, so there are different. The leukocytes or the white blood cells increase in different conditions when you are infected, and there are different types of them, which can be very informative about the type of pathogen which is infecting particular individual. Okay, for instance, monocytes or macrophages are different cell neutrophils. They can inform you about different types of infection, and their number also. So for instance, leukocytosis, it is above uh, 5 to 10,000 leukocytes in per ml of your uh, blood when you examine it under the microscope, or even more in case of leukemide reactions. Sometimes leukocytosis and leukemide reactions, which are characterized by increased number of the leukocytes in the blood, are confused with leukemia. So leukemia is a different thing. The nature of the cell changes. They also the number increases, but the nature also changes, which is distinguished by pathologists. Serodiagnosis is not useful actually. PCR and CD, using the CSF fluid, if you can extract, DNA from it and do PCR, then that is also a good technique for diagnosis. The treatment is not satisfactory treatment. No satisfactory treatment is available right now, and this antifungal amphotericin B is the drug of choice. Is inhibits the pathogen by binding to its cell membrane sterols, thus leading to cell membrane disruption and pathogen death. That is its mechanism of action, but uh, uh, it's also not very successful if you are if once the symptoms have started appearing. That means that the disease is already at advanced stage. It would not be possible for the drug to be administered there effectively, because many drugs, they do not cross the barriers which are there, the blood-brain barrier. So there are treatments using some kind of new devices which can cross this cribriform plate, and they also claim that stem cells can also be administered through this particular route. So some success, limited success, has been there in administration of different drugs against this to the brain tissue, but overall prognosis is very poor, and these are other drugs that are being used for this condition. And the prevention of this is that keep your head above water, wear nose plugs if you are a diver, you're swimming, avoid swimming in warm fresh water. uh, it has been seen that this particular parasite is abundant in warm, fresh water. That's why you should avoid these things. And chlorination of swimming pools and public water or public water supplies, that is also very essential to get rid of this particular problem. It's everywhere. Even in Pakistan, several cases have been reported. This is a report in the Lancet Infectious Diseases, reporting Naegleria fowleri in Pakistan, in Karachi. It's very strange that the water there is saltish and it's not suitable for Naegleria survivors. Still, you are having this particular infection in uh, there. And these are different parts of the world, the blue and other parts you can see. Uh, Pakistan is already there among the heavily affected areas, are prone uh, to have these conditions. These are different tropical or subtropical regions of the world where you can find this particular infection uh, prevalent there. So thank you very much for your uh, time, and we would discuss other free living amoebas um, in our lectures. You would get these slides as well, so you can just go through them and find out some research papers about this particular Naegleria fowleri, and hope to see you next time. Thank you very much for your time.